Hello students, I am Dr. Moinak Mukhopadhyay. We are going to learn today the topic that is glycolysis. That topic is there in your syllabus, I believe from class 9 onwards. I think it may be in class 9 or class 12 or 11, followed by these things are there in the syllabuses. But today I am going to discuss in the detailed way that how this glycolysis actually performs and what is the importance of this glycolysis. So in this class, in this particular video or in this particular channel, we are going to discuss several related topics. So if you are new to this channel, please kindly subscribe the channel and click the bell icon so that whenever I am going to post a video, that will directly reach to you. And if you already a subscriber to this channel, please do comment if you have any question in your mind. Okay, I will definitely answer all those comments and please like the video. So let's start today's discussion. What is the content of this lecture? The contents are divided into four parts. Number one, importance of glucose. Before going or before jumping into the whole lecture about glycolysis, we need to understand what is why this glycolysis is so important. And on this respect, we also need to know that the basic component, which is the major fuel of this glycolysis, this major fuel is glucose. And what is this? What is the importance of this glucose? Second, major pathway of glucose utilization, that in different pathway which is available for glucose utilization processes. What are those different pathways? That we also need to understand. Third one is the glycolysis overview. And finally, we talk about every step of glycolysis. Right? So let's jump into the content. At the very beginning, we all know glucose is a particular molecule which is considered as an energy source. So what do you mean by energy source? Energy source means if we consume that glucose molecule, that will enter into our body and by the process of metabolism, this glucose will convert into energy. Okay, fine. So now if this glucose is a main energy molecule, and we all know that glucose are mainly synthesized by which organism? That is plant, right? By the process of photosynthesis. Okay, so it is a major component or major food source of plant, and also all other kind of animals are mainly depend on this glucose. Whatever the organisms are, that doesn't matter. It may be a eukaryotic organism, it may be a prokaryotic organism, it may be a bacteria, fungi, every organism. Whole energy chain of the living system is based on this glucose. So we can say that this glucose is a central part of the metabolism. That is why it is very important to know the metabolic pathways which is related to this glucose. Now, it is rich in potential energy. Where from this potential energy come from? What is your idea? Where from this potential energy come into inside this glucose? As I have already told just a few minutes back, that glucose is synthesized by which organism? Plants, by the process of photosynthesis. And in this particular process, solar energy is being converted into chemical energy. That conversion potential enriches the glucose with a huge amount of potential energy. Okay, so that is why glucose is considered to be a fuel. And also in every organism, as I've already mentioned it, as every organism, there is a metabolic chain, and every metabolic chain that is basically based on the glucose. Now, the third point is, if glucose completely oxidizes, it will produce carbon dioxide, water, and energy. Again, if we consider the pathway of photosynthesis. Here, carbon dioxide and water combined with each other with the help of sunlight to form what? To form glucose. Now, that same glucose, when undergo the process of metabolism, it degrades and it produces free energy along with the same thing which are involved for the production of glucose. What are those things? Carbon dioxide and water. And the fourth point is it is a precursor. What is the meaning of precursor? Precursor meaning the particular point from where different pathways can begin. That is known as precursor. So just look at this particular structure. If we consider glucose as a central molecule, glucose have variety of role in the total living organism world. That is why they are considered as a central molecule. They are central molecule, glucose. They have multiple array of function. Number one, they can be stored as storage molecule like glycogen, like starch, like sucrose. Glycogen is a storage component of whom? I think you all know the answer. Glycogen is a storage component of animal. Similarly, plant is a storage component of starch is a storage component of plant. Okay. Now, this glucose is actually producing or taking a very good role for the production of structural polymer. What is structural polymer? We all know that if we consider plant have a cell wall. A cell wall is a structural component of a cell, right? So cell wall is made up of, plant cell wall is made up of what? Mainly cellulose. Cellulose is a polysaccharide, which is a homopolymer of glucose. That is why different extracellular matrix and cell walls are made up of glucose or glucose derivative. What is the meaning of glucose derivative? A component which is made from glucose. That is known as glucose derivative. The fourth one is production of components which are essential for living organism. Like what is your understanding based on this particular component? Living components which are essential for living. The meaning is that, let's say a component here it is mentioned is ribose 5-phosphate. Ribose 5-phosphate is important for the production of nucleotides. So nucleotides means nucleotide is a component which is present in DNA. Right? It's a DNA unit, nucleotide. So glucose is also required for the production of ribose 5-phosphate via the process that we are going to discuss in somewhere later in our different classes. That is pentose phosphate pathway. Okay. So now the fourth one, which we are going to discuss here in this particular class, that is the oxidation of glucose via glycolysis, which produce pyruvate. But this oxidation is not a complete oxidation. In glycolysis, glucose is not completely oxidized. It is partially oxidized, and complete oxidization requires the following steps like TCA cycle, like electron transport and oxidative phosphorylation, and followed by energy generation. We are going to discuss every detailed steps or detailed pathways here only in this channel. So that is why please stay with us here. And if you like this video, please share it with your peer group. So now we start the process glycolysis. So now you can easily able to understand why this glycolysis is important, because this is a key source of energy generation process for a huge group of organisms, and it comes with the letter glycis, which means sweet or sugar, and lysis. Lysis means it's splitting, it's cleavage. Lysis of sugar, that is known as glycolysis. Here, this is a enzymatic process. What is the meaning of that? That this whole pathway is controlled by enzymes. As you all know, enzymes are biological catalysts. Here, I'm also going to discuss about enzymes in my subsequent videos. So, enzyme, this is a process which is driven by enzymes. And Subsequently, it produces pyruvate. So, among the whole array of pathways, all different pathways, this particular pathway is discovered first. So that, and this is also the most understood pathway still now in our biological system. So in this particular pathway, before we go much more inside into this particular pathway, you just need to know a few little things. Number one, this particular pathway is important because whatever an organism are, the meaning is whether an organism is aerobic, whether an organism is anaerobic, doesn't matter. This pathway is present in every organism, whether it's a eukaryotic or prokaryotic, whether it's aerobic or anaerobic, every organism have this particular pathway. That is why this particular pathway is very, very, very important to know. That means aerobic means presence of oxygen, anaerobic means absence of oxygen. That is why this pathway is known as oxygen independent. Now, this is an important pathway for everyone, those who are living, and for every organism. Glucose is the main source of energy. That is why it is very important to know how this glucose is being converted to final form of energy. This pathway is divided into two parts. First one is the preparatory step. That means preparation is required for the oxidation of glucose. And the first step, which comprises of 5 reactions, is the preparatory step, which contain one priming reaction. I will discuss in detail what is the meaning of this priming reaction, second priming reaction, and a cleavage reaction. In the same way, the phase 2 is the payoff phase. Payoff. What we are getting from that pathway is there in this second phase. So first one is the preparatory phase and second one is the payoff phase. The preparatory phase contains 5 steps. Payoff phase also contains 5 steps. Payoff phase actually deals with the oxidation part. The oxidation part, ATP formation part, direct ATP formation part based on substrate level phosphorylation as well as second ATP formation part. Here we are going to see that another molecule is being involved in this particular process. Another molecule is not directly involved, it's being produced in this particular process, that is NADH. Okay, and final end product is pyruvate, which is a 3-carbon compound. Here every structure is mentioned in the right hand side. Okay, so let's start with the hexokinase. Hexokinase is an enzyme which is required for the first reaction. As I have already told you, the name of the reaction is priming reaction. Priming means marking something with what? With a phosphate. Okay, so with the help of this particular reaction, where one molecule of glucose in the presence of ATP and by the use of the catalysis of hexokinase enzyme produce glucose 6-phosphate, one ADP and one hydrogen plus. This is a irreversible reaction. That means glucose 6-phosphate cannot be converted to glucose. It is not possible, right? So now, why this step is important? Why this particular step is important, that formation of glucose to glucose 6-phosphate? Glucose, if you look at its structure, it doesn't contain any charge. So now, when glucose enters into the cell via different carrier protein, if that glucose is not utilized, that glucose will leave out from the cell because it doesn't have any charge. Okay, so now the first thing is to trap the glucose inside the cell. So for trapping of glucose inside the cell, it requires a tagging. Tagging of what? Tagging of phosphate. Phosphate comes from ATP. That is why energy is being utilized in this reaction. Now, the second reaction is fructose 1,6-bisphosphate generation from glucose 6-phosphate. So this is a 2-step process. Step number 1, glucose 6-phosphate is first converted to fructose 6-phosphate. And step number 2, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Okay? So let's discuss step number 1. The step number 1 is conversion of glucose 6-phosphate to fructose 6-phosphate. Or I can assume that you have an idea that glucose and fructose are isomer of each other. Their structural formula is different, but functional formula is same. That is C6H12O6. They are isomer of each other. So we just require an isomerization reaction for the conversion of glucose 6-phosphate to fructose 6-phosphate, right? But here, one thing you need to know, that is glucose 6-phosphate is present in ring form, and final product of fructose 6-phosphate is also present in a ring form. But isomeric conversion is not possible until and unless it converts into an open chain conformation. Because here, as we all know, glucose is contain a aldol group, CHO, but here fructose contain a keto group. So keto aldose to ketose conversion is not possible without opening the ring. That is why ring opening is a part. That means ring structure glucose first convert into open chain form. Then open chain glucose 6-phosphate is being isomerized to form fructose 6-phosphate, and then fructose 6-phosphate is again converted to ring structure of fructose 6-phosphate. The enzyme which is required here, phosphohexoisomerase. So phosphophosphate containing hexose isomerization process, right? So in overall, you can say that it's a phosphate containing hexose isomerization process. I don't know. That is why the name of the enzyme is phosphohexoisomerase. So, and the second step involves another set of phosphorylation that is based on kinase enzyme, which is again an irreversible reaction because that involves ATP for a substrate level phosphorylation. And the enzyme which is important here is phosphophotokinase, one of the most important enzyme in glycolytic pathway. Yes, phosphophotokinase is the most important enzyme or most important regulatory molecule in this particular glycolysis process. So similarly, as of the first reaction, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Here bis means two phosphates are associated in two different carbon. If they are associated simultaneously, like this one, it may known as diphosphate, like ADP. ADP is adenosine diphosphate. Two phosphates are attached with each other. But bisphosphate means two phosphates are attached separately into separate carbon atom. Okay. So now you may have an idea that why should we invest energy for the sake of energy? But you should know that if you want to gain something, you have to invest something. If you want to gain knowledge, you have to invest time for reading, for listening, right? So similarly, if we want to extract energy, we have to use that to make a process irreversible. Until and unless the process is irreversible, it will not ensure for the production of energy, right? So the next step is the cleavage of 6-carbon glucose into 2 3-carbon fragments. So glucose is a 6-carbon component. So when it gets cleaved via an enzyme aldolase, it will produce dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So which is being cleaved? The cleaved thing is fructose 1,6-bisphosphate. So here in two different color, it is mentioned about the position of molecules and the production of molecules. So fructose 1,6-bisphosphate is being cleaved in this particular position after third carbon atom, and it produced dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and both are precursor of carbohydrate molecule. Okay, so the isomerization of the 3-carbon phosphorylated sugar, that means this dihydroxyacetone phosphate directly doesn't have that much access towards the metabolic pathway. That is why it should convert into glyceraldehyde 3-phosphate. It should convert into glyceraldehyde 3-phosphate by the help of an enzyme known as triphosphate isomerase. So in this way, more or less 90% of the component which are there, those all components are cleaved down. Okay, so now we are going to enter into the payoff phase. It is payoff. That means we are going to get some energy either in form of direct ATP or in form of some high energy molecule. The example of high energy molecule is NADH, FADH2. They are the example of high energy molecules. So now the sixth reaction, this is the sixth reaction catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. The sixth reaction, glyceraldehyde 3-phosphate, with the help of one NAD plus and phosphate, converted to 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate plus NADH plus hydrogen. Okay, so glyceraldehyde is first converted to 1,3-bisphosphoglycerate. If you have time, if you want to understand the inner mechanism for this particular reaction, you can discuss in different books, but this particular reaction occurs in two different steps. Step number one, glyceraldehyde 3-phosphate first being oxidized into the acidic component. Okay, into the acidic component. Now, this acidic component is being phosphorylated to form 1,3-bisphosphoglycerate. So now, here comes the first step where we are going to get some ATP, the real ATP, 1,3-bisphosphoglycerate by the help of an enzyme kinase. What kinase or which kinase? Phosphoglycerate kinase. It is converted to 3-phosphoglycerate plus ATP is being produced. So now just think about, from one glucose, which is 6 carbon, 2 3 carbon components are there. Okay, so from here one ATP is produced. One ATP will produce from that particular group also. Okay, so now through this reaction, 2 molecules of ATP will be produced because of the presence of 2 molecules of pyruvate to 2 molecules of glyceraldehyde 3-phosphate, 2 molecules of glyceraldehyde 3-phosphate followed by 2 molecules of 3-phosphoglycerate and 2 ATP. Okay, so now the 3-phosphoglycerate will undergo a process of 3 different steps for the production of another molecule of ATP and its final product, that is pyruvate. 3-phosphoglycerate, by use of an enzyme that is called mutase, is formed 2-phosphoglycerate component. What is the difference between 3-phosphoglycerate and 2-phosphoglycerate? The phosphate group is transferred from third position to second position. That is why it is 2-phosphoglycerate. Fine. Second one is the second reaction is the conversion of 2-phosphoglycerate to phosphoenol pyruvate, followed by an enzyme name is enolase. The name of the enzyme is enolase, but it can also known as hydrolase enzyme. So it introduce one double bond inside the component. Okay, so 2-phosphoglycerate then converted to phosphoenol pyruvate by using an enzyme that the name is enolase, which is an hydrolytic enzyme. So this phosphoenol pyruvate contain one unsaturation, and this phosphoenol pyruvate finally being converted by pyruvate kinase enzyme by producing again ATP and pyruvate component. So ATP is a very important thing. It's energy. Here also 2 ATP is being formed. Just look at that particular thing. So 1 glyceraldehyde 3-phosphate, 1,3-bisphosphoglycerate. So that means 2 1,3-bisphosphoglycerate. So 2 3-phosphoglycerate, 2 3-phosphoglycerate, 2 phosphoglycerate, 2 phosphoenol pyruvate, and 2 pyruvate. So every component having a 2 atom, two components are there. Before concluding the class, let us calculate the total amount of energy that is being produced followed by using this glycolysis pathway. So the amount of energy which is being produced is total 2 ATP plus 2 ATP plus 2 NADH. 2 ATPs are being produced, followed by 2 ATP, followed by 6 ATP, 6 equivalent ATP that directly comes from NADH. So total how much ATP is being produced? 10 ATP is being produced. And how much ATP is being utilized? -2. So what is the net gain of ATP in this particular process? That is 8. Fine. So this is the end of our today's video, and we have discussed the glycolytic process, energy, and the number of energy ATP which is being generated via this particular process, the importance of glucose, importance of glycolysis, etc. So if you really like this video, please do subscribe my channel and hit the bell icon so that whenever I will post this kind of educational video, you will directly get notified. Okay. And the thing is that if you are not able to understand any particular point, let me know through the comment section and I will definitely help you out. Thank you so much.